Welcome to the second part of the presentation, and we will be discussing oxygen toxicity, how it relates to seizures, and how a ketogenic diet may prevent those seizures. So you're probably thinking, why are we even talking about oxygen toxicity? Well, once we understand what oxygen toxicity is, then we'll be able to understand why it's important. So what is oxygen toxicity? Well, the toxic effects of oxygen. So this occurs when partial pressures of oxygen are elevated, and this is termed hyperoxia, causing oxygen radicals to form in excessive quantities. So oxygen radicals are kind of a free radical, and so they wreak havoc just like any other reactive oxygen species. Also, what occurs with oxygen toxicity is enzymatic and cellular dysfunction, which is caused by the hyperoxia. And so when this occurs, organ systems can be damaged or dysfunctional, and some of the organs include the brain, eyes, liver, and lungs. They can all be affected. So oxygen toxicity has been linked to complications seen in divers and other persons exposed to hyperbaric oxygen. And the divers could be also exposed to the hyperbaric oxygen when they are under treatment for decompression syndrome. And so the major complication here that we're looking at is oxygen toxicity, seizures, and we will see why this is important in just a bit. So let's discuss why oxygen toxicity is so important. Well, the potential outcomes for divers, especially Navy divers. The outcomes can include convulsions or seizures, unconsciousness, and death. The importance of understanding oxygen toxicity rests on the potential outcomes of exposure to hyperbaric oxygen. Becoming unconscious and/or convulsing while on an excursion is extremely unfavorable and may result in a failure or casualty. This is a potential outcome of acute oxygen toxicity. So as it relates to divers, acute oxygen toxicity may occur if the gas mix or rate of exposure to the mix is not appropriate for the depth of the dive. According to Herbert Newton, MD, Associate Professor of Neurology at Ohio State University Medical Center, he states, although uncommon, generalized seizures at depth are often fatal. This is quite important. And these effects, or the outcomes because of the effects of oxygen toxicity, may be preventable through a ketogenic diet. So what are the statistical data regarding diving accidents and deaths? Well, first, reporting diving accident data is voluntary and is only based on a small sample size according to Divers Alert Network, DAN, D-A-N, which makes it difficult to ascertain accurate statistical information regarding diving accidents and deaths, especially the underlying cause of morbidity and mortality. Nonetheless, statistical estimates exist which demonstrate a yearly occurrence of roughly 1,400 injuries per 1 million divers, and for every 10 accidents or injuries, there is one death. So what are the factors related to diving accidents and deaths? Well, inadequate training or experience. 50% of accidents or deaths occur in divers with less than 2 years experience or less than 60 total dives. Panic is more likely within this group because of the inadequate training or experience. And I quote, "Panic kills." We will see shortly how panic can lead to oxygen toxicity and its complications. Equipment problems are a factor. Gas mix or rate of exposure to the mix is inappropriate for the depth, as we just mentioned. Thus, it is imperative to make sure dive tables are carefully interpreted and mathematically confirmed for the upcoming dive event. Now, 41% of fatal accidents are related to running out of gas, so that may be an issue as well. Environmental causes are factors as well, including current, visibility, surface conditions, and overhead barriers. This may lead to panic and actions that lead to death. Finally, host factors are also associated with diving accidents and deaths, which relate to fitness, obesity, asthma, and cardiorespiratory disease. So decreased fitness, increased obesity, obviously asthma and cardiorespiratory disease are also unfavorable situations. So where does oxygen toxicity fit in though? Well, oxygen toxicity and its outcome may be uncommon, but when it occurs, its significance becomes apparent. If there are a means to mitigate or at least reduce the risk of seizure caused by oxygen toxicity, then why not consider oxygen toxicity when discussing the safety of diving? Regardless of the prevalence and incidence of oxygen toxicity seizures, focus should still be had on limiting the occurrence of such events, especially if serious consequences may result. After all, insurance policies will not cover you for diving because it is a very high risk kind of recreation. So why are the effects of oxygen toxicity important to understand? Well, because it makes divers and other personnel aware of the effects of such an exposure. And this exposure can obviously be dangerous. So once the effects are understood, then we could possibly prevent the outcomes. So what are the effects? There are acute effects, which has been termed the BERT effect. Central nervous system effects make up the BERT effect. And chronic effects, the Lorraine effect, which are typically respiratory system effects. But for this section, we'll be focusing on the acute effects. So what is the BERT effect? We just mentioned that it was the CNS effects of acute oxygen toxicity. But with this effect, though, there comes peripheral vasoconstriction, which can lead to facial pallor, which might be a sign of the acute oxygen toxicity. Diaphragmatic twitching may occur, which leads to cogwheel-type breathing patterns. Continued exposure can lead to vertigo and nausea, which can eventually lead to altered behavior and clumsiness, and then potentially convulsions, which are tonic-clonic in nature, with a possible neurogenic pulmonary edema. A commonly identified sign of acute oxygen toxicity is lip twitching, which can precede a seizure. And there are aggravating factors of CNS toxicity, and they include increased partial pressure of carbon dioxide, stress in general, cold, and deficiency in selenium, zinc, and magnesium. Part 4 of this presentation will discuss nutritional strategies related to oxygen toxicity seizures and also the ketogenic diet. Of importance here, though, the increased carbon dioxide should be pointed out real quickly. And we all should know that increased carbon dioxide will increase with physical activity, and so it's possible that it could also increase with the metabolic changes associated with the ketogenic diet. Because as we saw in part 1, when acetoacetate is decarboxylated to acetone, it yields a carbon dioxide molecule. And so I asked the question, could this portion of what the ketogenic diet can do create increased vulnerability to oxygen toxicity seizures? It's possible. So I had to let you all know that. So how does oxygen toxicity affect the central nervous system? Well, in general, oxidation and polymerization of sulfohydryl groups of enzymes occur, leading to cellular damage. As oxygen concentrations increase, this can lead to extreme amount of free radical concentration, which could potentially overwhelm the antioxidant defense networks. Continued exposure could cause cellular metabolic alterations and or enzyme inhibition. And so inhibition of glutamic acid decarboxylase is possible. And this enzyme GAD, is responsible for converting glutamic acid or glutamate into GABA. And this conversion also necessitates pyridoxal phosphate, which is the active form of vitamin B6. So there's another nutritional implication there. And so with oxygen toxicity, GAD is inhibited, and so this will lead to decreased amounts of GABA and increased amounts of glutamate. And so increased glutamate concentration can lead to increased amount of reactive oxygen species due to the high glutamate concentrations. And we saw that at the end portion of part 1 of this presentation. So we saw that deficiency of magnesium, zinc, and selenium may exacerbate central nervous system toxicity. So are there any other nutritional deficiencies related to oxygen-induced seizures? Well, copper is, and decreased copper can lead to the dysregulation of genes involved in cellular responses to oxidative stress. Also, with low copper concentrations, impairment of copper-mediated NMDA receptor function can occur, which can lead to increased neuronal excitability and excitotoxic neuronal injury. If the impairment of copper-mediated NMDA receptors occurs, this may predispose to cascades leading to seizures and epileptogenic events. So that's very important to consider. Now, decreased copper also can lead to decreased dopamine, beta hydroxylase, and cytochrome C oxidase. And so this enzyme, dopamine beta hydroxylase, can lead to the decreased conversion of dopamine to norepinephrine, which could possibly lead to dysregulation of autonomic nervous system associated with the cardiovascular and other body systems. Now, although it is known that severe deficiency of copper, also called Menkes disease, will result in these scenarios, it is wise to consider marginal deficiency of copper as a possible exacerbating factor in oxygen-induced seizures. Obviously, copper levels will not be so low that a lot of these things will occur, but if copper concentrations are low in the body, there is a possibility that it could be exacerbating because of the association of what copper is for. So there's more to the story when it comes to the central nervous system effects in relation to oxygen toxicity, and that's the latent period. This latent period could last from minutes to hours to days, and essentially it is generally unpredictable, and especially unpredictable on an individual basis. So the more consecutive days of diving or prolonged periods of hyperbaric oxygen, the greater the likelihood of symptom presentation. And so having too high a partial pressure of oxygen at too deep of a depth will increase the risk for oxygen toxicity. And the risk fluctuates throughout the day and is different from day to day within the same individual. These two facts that we've just understood here actually might make dive tables irrelevant or maybe even invalidate them. And there are factors that actually modulate the latent period. They include increased carbon dioxide, stress, exercise, general physical exertion. And so each of these factors shorten the latent period, meaning symptoms of CNS toxicity may occur sooner than later. Carbon dioxide and exercise both drastically decrease the latent period, and light has been found to modulate the sensitivity to oxygen toxicity. So darkness can shorten the latent period. So just to briefly mention seizures, we should look at the ones associated with oxygen toxicity, and those are generalized seizures. And the causal factor is the same. It's lipid peroxidation of cell membranes and consequential neuronal damage. These generalized seizures are evoked from deep brain regions, and they spread to both hemispheres of the brain. And it also includes the frontal lobe. The tonic-clonic seizure is essentially alternating muscle tension and jerking. And this may last up for 20 to 120 seconds with a post-ictal state that may last up to 15 minutes. And so the post-ictal state is essentially the time period after the seizure. And during this time, the victim is unaware of what happened. So can seizures result in any kind of other pathologies? And we mentioned that neurogenic pulmonary edema is possible, and this is fluid accumulation in the lung, which can occur during the post-ictal phase. The pathogenesis of NPE is related to a CNS discharge, which is related to the actual seizure, and this leads to an increase in sympathetic nervous system tone and catecholamine release. And then that leads to an increased pulmonary and vascular resistance and cardiac contractility along with tachycardia. And so that eventually leads to an increased pulmonary vascular pressure, which results in alterations in Starling's forces, and damage to the pulmonary basement membrane occurs. And so the ultimate endpoint of this is that there is extravasation of fluid into the lung interstitium. Also, myocardial stunning can occur, which is stress placed upon the heart, which is due to the increased pressure within the pulmonary vasculature due to the NPE. And so the myocardial stunning may occur due to the acute stress placed on the heart, as well as the increased pressure in the lungs from the overt fluid accumulation. Inflammatory cascades are possible as well, and this activity may increase the severity of the NPE and myocardial stunning. So injury from oxygen toxicity and seizure leads to the expression and release of pro-inflammatory molecules, and these are thought to enter the systemic circulation and cause inflammatory associated changes in the lung, resulting in a translocation of fluid, the extravasation. And then epithelial cytokine expression occurs, and the release occurs as well due to the mechanical insult from the increased pulmonary resistance and pressure. And this also leads to extravasation of fluid. And there's also sympathetic nervous system activation, and the activation is related to the tone increase due to the seizure. And so within the CNS, this increased sympathetic nervous system may inhibit neurogenesis in the dentate gyrus of the hippocampus. So this illustration essentially sums up everything that we've talked about up to this point. And the major point, though, of this illustration is that the decreased latent period will occur either directly or indirectly based on the factors on this illustration. And ultimately, they could result in oxygen toxicity seizure and potentially injury or death. So we just looked at oxygen toxicity and how it might affect divers in that it may cause seizures. But now we're going to take the ketogenic diet and we're going to see possibly how it could prevent those seizures. So the ketogenic diet could potentially suppress or diminish the expression of pro-inflammatory molecules, limit the effects of the sympathetic nervous system activation on neural structures, fortify antioxidant defense networks, increase GABA concentration, protect against glutamate receptor agonists and other excitotoxins, and resist metabolic stress. These are all going to be a review of what we discussed in the first part of this presentation and also what we just covered in oxygen toxicity. Hence the reason for the title, Connecting the Dots. So up first is the suppression or diminished expression of pro-inflammatory molecules. So the ketogenic diet essentially inhibits inflammation, which prevents the unfortunate fate of oxygen toxicity, seizures, and its complications. So the fatty acid load provided by the KD will hopefully activate the PPAR alpha that we talked about earlier, and that will lead to an inhibition of transcription factors like nuclear factor Kb and activation factor or activation protein 1, and that will hopefully suppress any inflammatory cascades. And so seizures have been associated with inflammation, so if we can curb the inflammation with the ketogenic diet, then potentially we could prevent the seizures. And if there are subsequent events that occur because of the seizure, possibly we could decrease the severity of those as well. So this illustration essentially shows how fatty acids can create changes in genetic expression. So once the fatty acid enters the cell, it can bind to the PPAR alpha, and once it's activated, the activated receptor then enters the nucleus to complex with the retinoid X receptor, which causes a site-specific DNA binding, and this binding may lead to decreased transcription of genes related to inflammation. And so remember, while on a ketogenic diet, along with ketones in the blood, there are also lipids, and those include free fatty acids. So there may be limitations of the effects from the sympathetic nervous system activation on neural structures. And so this activation is related to seizures, and it can essentially cause disturbances within hippocampal neurons. And since hippocampal neurons are associated with memory fabrication, there may be some sort of connection between the two when it comes to the confusion or forgetful state that follows the seizure. So the KD may offer resistance to the metabolic changes due to the increased amounts of glutathione peroxidase within the hippocampal regions, as well as increased uncoupling proteins in the mitochondria. These are two things that we mentioned in part 1, the glutathione peroxidase increases and the uncoupling of the proteins in mitochondria. So this may all be afforded by beta hydroxybutyrate. And so essentially there's going to be an increase in antioxidant concentrations, which we'll get to, and decreased reactive oxygen species. And so the energy pathways that may be enhanced too could help this situation out as well. So antioxidant defense networks may be fortified by KD, and this is important because increased oxygen concentration can result in a free radical concentration that may overwhelm antioxidant defenses. And ketone bodies have been shown to decrease the amount of coenzyme Q semiquinone, which helps prevent the production of reactive oxygen species. Also, we just mentioned that glutathione peroxidase is also increased by KD in the hippocampus of rats and uncoupling proteins in the mitochondria. This also helps lead to decreased ROS production. Increased GABA concentration is important because we mentioned that the oxygen concentrations, when they are very high, they inhibit GAD, which is responsible for converting the glutamate into GABA. And so essentially the KD helps increase GABA concentrations. And this GABA-mediated inhibition, or this GABA concentration, is thought to help with inhibition. And this inhibition is thought to be neuroprotective. And make sure that we recall that GABA has functions related to synaptogenesis, which is the creation of synapses between neurons, neurotransmission, which helps with the propagation of information in the nervous system, and overall neuronal protection. Glutamate receptor agonists may also create some problems. And the increased glutamate concentration, we said, originates from the oxygen-inhibited glutamic acid decarboxylase, which we just looked at. And so we should note that the excitotoxicity causes neurodegeneration and is implicated in many neurodegenerative diseases. And so acetoacetate is thought to protect against toxicity induced by excessive glutamate, which we mentioned in part 1 of the presentation. And the KD is protective against apoptotic cell death caused by glutamate receptor agonists and excitotoxins. And so during the excitotoxicity, there is an influx of calcium ions. And so this influx is thought to activate many calcium-dependent enzymes, many of which are degenerative in nature, like caspases, which are a family of proteases associated with apoptosis, necrosis, and inflammation. And so we talked about this in part 1 as well, and that the KD increases calbindin, which helps to buffer intracellular calcium. And we just mentioned the things that are possible when calcium increases in the cell. Clusterin was mentioned earlier as well, and this is a pro-death signal that is activated by excitotoxins. And so the KD actually can decrease this pro-death signal, which would be helpful, especially if excitotoxins are increased in these situations. The effects of metabolic stress may be dampened. And so with carbohydrate restriction, there will be a decreased availability of glucose, which leads to the induction of stress proteins. I'm sure you've heard this and seen this before. And so with the decreased amount of reactive oxygen species that occurs and the stabilization of intracellular calcium and the maintenance of mitochondrial function, this allows for an increased amount of handling of metabolic stress via decreased inflammation. And so we mentioned earlier that the mitochondrial biogenesis and ketosis can lead to neuronal resistance to metabolic stress. And our favorite molecule of choice here is beta-hydroxybutyrate, and it can possibly do so by restoring substrates for mitochondrial electron transport chain and bypassing mitochondrial complex 1 via production of succinate. Now, beta-hydroxybutyrate also improves cardiac energy utilization and efficiency, like we talked about, by 25%. So could the KD potentially offer cardioprotection during times of increased strain, such as the events associated with seizures? Some studies suggest that the KD can improve energy uptake in heart failure patients. So this is a possibility. So the metabolic stress and the overall stress that occurs from oxygen toxicity could be at least lessened by the ketogenic diet. So overall, what is the big connection here? Essentially, a KD may act as a buffer to increase the latent period. If we increase the latent period, that means that the symptoms of oxygen toxicity will not occur in such a quick fashion. It'll take longer for that to occur. This might be a good thing because by the time the symptoms show up, the diver may be out of the water, and it would be much better to have a seizure outside of the water than while in the water. So the latent period may be increased because of the resistance to metabolic stress, the fortification of antioxidant networks, the suppression of pro-inflammatory molecules, the regulation of neurotransmitters. And so if we look at all of those possibilities and we say we could increase the latent period, this is a good thing. So honestly, why not at least employ a KD for its favorable effects, especially since the severity of any adverse effects are very minimal and unlikely. So who knows? It could save a life or two. And up next, we'll be discussing human performance and how a ketogenic diet could affect it.